All right, everyone, let's take a look at our tissues that we need to know for the first lab exam. Now, you may be wondering just where the heck do I start? And so the first place to start is to be able to identify the tissues. That's going to be what most of the tissue questions are about, just identifying. And then there will be additional questions, a couple of them on what is the function of this tissue or where is its location. But you have to be able to tell what the tissue is that you're looking at first in order to be able to identify its function or its location. So let's just start with, can I tell which category of tissue that this particular image belongs to? So it might be epithelium, connective, muscle, or nervous tissue. If it's epithelial tissue, we're looking for some specific characteristics. One, the cells will be tightly packed together. Two, you will see some sort of an apical surface because these cells line other body parts or they surround lumens, tubes that are moving through the body. So that apical surface will be the free surface next to the empty space, the opening, the cavity, etc. We're going to have a basal surface, and at that basal surface we'll see the basement membrane where that epithelium connects to the underlying connective tissue. For connective tissue, on the other hand, the characteristics we see here are that the cells will be spread apart. We'll see extracellular matrix, and we will often see fibers in that connective tissue. For muscle, the cells are also tightly packed, similar to what we saw with epithelium, but no apical surface here. That muscle tissue is going to be adjacent to other tissue types, like connective tissue, for example. And then finally, for nervous tissue, we're looking for large cells, that would be our neurons, and they will have multiple processes or extensions of that cell that extend away from that nervous tissue in that slide. So when you come to the lab test next week, you're going to see microscopes set up and pictures printed out, and you need to be able to step up to that image or that microscope and quickly decide what kind of tissue you're looking at. So here's a quick little step-by-step that I would recommend. Number one, do you see an apical surface? Do you see an empty space, a white space next to the tissue that you are being asked to identify? If yes, you're looking at an epithelial tissue, and if not, it's one of the other three categories. So let's look at those three categories. How do we tell those apart? So the next thing to look at if there's not an apical surface is to determine if the cells are spread distantly, or maybe you can see fibers, or maybe you can see lacunae. That would tell you it's one of the connective tissue types. Otherwise, you might be looking at muscle or nervous tissue. If the cells are tightly packed together, but there's no apical surface, there's no empty space, like a lumen or an open cavity, then it's probably going to be muscle. And then for nervous, you're looking for large cells with extensions extending away from them. All right, so let's go back to step one. Let's imagine that you come to that slide and you see that there's an empty space, an apical surface somewhere, and you notice an epithelium. Layers of cells there are, because remember that's the first part of the name for epithelial tissue, and then we'll have to determine the cell shape. So, are there multiple layers of cells? Do you see rows of nuclei or rows of cells stacked upon one another? If you do, it is a stratified epithelium. If you see just a single layer of cells between the apical surface and the basement membrane, then it is a simple epithelium. Second thing to do is determine the cell shape. And the cell shape will either be squamous, cuboidal, or columnar. So for squamous, remember they're squished and flat. Cuboidal are cubes, so they look like squares in 2 dimension, and columnar are elongated like columns. If you have already determined that it's a stratified epithelium and now you're trying to identify the shape, remember that you must look at the shape that is at the apical surface, right? Cilia and goblet cells are only present in columnar epithelium, so if you see either of those features, you know that it must be columnar. Also for columnar, if there are hair-like extensions on the apical surface, make sure you include ciliated or non-ciliated if there are no hair-like extensions. So all of our columnar will want to add in ciliated or non-ciliated. Okay? If instead you've determined that you are looking at a connective tissue, the first thing to do is to see if you have any lacunae. Remember, the lacunae are the empty white spaces, the bubbles that house your cartilage, cells, which are called chondrocytes. So if there are lacunae, you are dealing with one of the three cartilage. If not, it's either connective tissue proper, bone, or blood. Okay? If you see those lacunae, you need to determine if there are visible fibers, because the only cartilage that we have that has easily visible fibers will be elastic cartilage. You can kind of see the fibers in fibrocartilage. I'll point that out when we get there, but those dark, easily visible fibers that we see in elastic cartilage are most readily apparent. All right, hyaline cartilage will have fibers, but they are hyaline or glassy, so transparent, and fibrocartilage has what kind of looks more like brush strokes, and so we'll point that out on that slide too. If you don't have lacunae, it's not a cartilage, but if you have visible fibers, then it's one of these categories. And I think this is where we kind of get a little bit bogged down in some of these step-by-steps, and it's easier to just take some of these other connective tissues one by one. But areolar, elastic, dense irregular, and reticular connective tissues all have fibers that are easily visible. Adipose, bone, and blood, those fibers are either not there or they're not easily visible. So bone's pretty easy. It just looks like tree rings. And blood, I think, is also relatively easy to identify. You'll see those red anucleate cells with blood. All right, so what I'm going to do next is go through each tissue type. I'm not going to do them in any particular order. I picked these at random. And what we're going to do is imagine that we're coming up to that slide on a test question, and we'll start going through our criteria, and I'll talk you through how we can identify it. All right, so we start with this one. This could be a potential question on your test. Identify this tissue. So let's look at some of the characteristics that we see. Right? I see a lot of empty space here. So it looks to me like there's empty spaces, cavities. So I'm probably dealing with something that has an apical surface, which tells me it must be an epithelium. Next thing I want to look at is how many layers of cells there are. So if I look at my rows of nuclei, it appears to be just a single layer of nuclei along these lines. So I think I'm dealing with a simple epithelium. And finally, I want to look at the shape of those cells. And if I look at the shape, those cells appear to be squished and flat, meaning this must be simple squamous epithelium. Okay, so start with that. The other thing to add on here, and there will be a few questions on function and location. Functions for simple squamous are filtration and diffusion. If you think about a filter, like a coffee filter, for example, very, very thin in nature, just like simple squamous, which is a thin layer of squishy cells. Locations, this particular location is the alveoli, which are in the lungs. And then for all of our for all of our epithelial types, you probably want to include the word lining or covering to get full credit. So we line blood vessels or line body cavities with simple squamous epi as well. Here's our next slide. So what I'm looking at here is there's some cells scattered around here, but this one really catches my eye. That's a big cell, and if I look at the extensions off those cells, there's multiple extensions. So this is our nervous tissue. Functions here include sensory input, integration, and motor output. Here's our next one. So what I'm looking at here is I see some lines that kind of all appear to be going in the same direction more or less. I see some nuclei, these sort of darker dots here and here and here and here, but they don't seem to be regularly spaced. My cells appear to be spread out. I don't see lacunae. Now I do see some empty white spaces, but they're not bubble-like in shape, and I don't see cells in those white spaces. So those would not be lacunae. What I do see are fibers, and these are the fibers that are running in a parallel direction. And that would tell us that this is dense, regular connective tissue. So we know it's a connective tissue because the cells are spread out. We know it's not cartilage because there are no lacunae. And because those fibers are packed tightly, it's a dense connective tissue, and the regular array tells us it's dense, regular connective tissue. Dense regular connective tissue provides tensile strength in one direction and is found in your tendons, ligaments, and aponeuroses. those fibers are collagen fibers all running in one direction, and collagen provides tensile strength, and therefore that's what this tissue does as well. This is one of our dense regular connective tissue slides. If this doesn't look like the one you looked at in lab, yours might have looked more like this. It's a slightly fainter slide, but you can still see those fibers that are all running in the same direction, and then nuclei of cells that are scattered through the tissue as well. All right, here's our next tissue. Let's see what we've got here. I've got a whole bunch of cells in here. I don't see an apical surface. This is my tissue here, and I would have an arrow pointing to this area, not this stuff down here, just this up here. So it's not epithelium. And I see lacunae. These are my bubbles, my white spaces that are going to be filled with my chondrocytes. So it must be a cartilage. I've got 3 cartilage options: hyaline, fibrocartilage, and elastic cartilage. And I see that there are some fibers in here. These dark lines, kind of all this dark stuff here, those are all my elastic fibers. So this must be elastic cartilage. Elastic cartilage provides strength and elasticity, and I think the easiest function to remember is elasticity because it's elastic cartilage. It's in the external ear and the epiglottis, and those elastic fibers convey the function to this tissue. Here's another one. Now, if I look at this slide, once again, although I do see some like white spaces, there's no distinct apical surface. So it's not epithelium. Right? Next, I do see cells that are relatively tightly packed. There's a cell and a cell and a cell and a cell and a cell. Cells all kind of packed together next to cells. So cells that are tightly packed with no apical surface, that category would be muscle tissue. And the next thing for muscle tissue is to decide whether there are striations, and the striations are the stripes that run perpendicular along the length of the cell. And I do not see any of those striations here. So it is a non-striated muscle, and the only of our muscles that lack striation is smooth muscle. You only need to know the functions of muscle in general: movement, heat generation, and posture. All right, we've got some more. Let's keep moving. Here's our next tissue, and on the exam, I would be pointing you to this area on the slide with our pointer. So I see that this tissue comes up right against this empty or white space. So that is going to be my apical surface of cells. Now I want to determine, since I know it's now epithelium, how many layers of cells do I have? So if I look at my nuclei, it looks like I've got one row and then another row stacked on top. I see the same thing over here. One row and then another row of nuclei stacked on top. So this must be a stratified epithelium. As for cell shape, if I look at the apical surface, those cells are a little more drawn out and elongated. I also see goblet cells. These lighter colored, almost white cells, those are all goblet cells. So it must be columnar, and therefore it is stratified columnar epithelium. Anything that is stratified, here's a little tip for you, all of the stratified epithelium have the function of protection. So you're only going to be asked for one function. If it's stratified and you put protection in there, you will be right on all of the stratified epithelium. Stratified columnar also secretes things. We find this tissue in the urethra and in the conjunctiva of the eye. Next tissue. So I'm looking right in this area, and again, you'd have a pointer on the exam to determine exactly where it is I want you to look. So I see cells that are scattered, but no empty space, no apical surface. It is not an epithelial tissue. And I see what looks like kind of bubbles or these empty pockets in the tissue. So those are my lacunae, and the purple dots in the middle there, those would be the chondrocytes. The background of this tissue is fairly consistent, and I don't really see any strings or fibers in here. That tells me that this is hyaline cartilage. And hyaline cartilage adds flexibility, support, reduces friction, absorbs shock. It is in the fetal skeleton, your nose, your epiphyseal plates, and the ends of your long bones. The fibers that are here are collagen fibers. You just don't see them because they are glassy. That's what hyaline means. So they're transparent, and you just get this diffuse background. This happens to be our weakest cartilage type as well. Right? Let's contrast this slide with our next slide. Okay, so here's our next slide. This tissue, again, no apical surface. I again see lacunae. There's my bubbles here and here with my chondrocytes in the middle. And sometimes you'll be able to see fibers. Sometimes they won't be that obvious. For fibrocartilage, which is what this is, the fibers look like brush strokes. And if I look here, I can kind of see what look like brush strokes in that cartilage. And you will often see sort of swirling teal and pink colors in that fibrocartilage as well. Fibrocartilage supports and joins structures together. It is found in the pubic symphysis, the intervertebral discs, and the menisci. And it contains collagen fibers. So fibrocartilage and hyaline cartilage both have collagen fibers. So if it's cartilage and it does not have the word elastic in it, the fibers are collagen. And fibrocartilage is the strongest cartilage. Here's sort of a zoomed out view of that fibrocartilage as well, so you can really see a little bit more of the brush stroke motion and some of the variations in color. Here's a tissue that I think is fairly straightforward. We see red or pinkish anucleate cells, no nucleus. Some white blood cells are visible here with those purple nuclei, so this is blood we're dealing with. And blood is for transport, regulation, and support. All right, let's take a look at this tissue. First thing, does it have an apical surface? And I see that it does. Here's our empty space. This would be our covering, our outer surface, or our internal cavity. And then this must be our apical surface here. So I know I'm dealing with an epithelial tissue. Next thing is, how many layers of cells do I have? So from the top here, from my apical surface, until I get to my basement membrane down here, where I see a transition to a different tissue type or connective tissue, I see there's lots of layers of cells. And the dead giveaway here as to what tissue type this is the apical surface with all this flakiness, all these sort of separated dead cells. That flaky apical surface tells me it is keratinized, stratified squamous epithelium. For all of our stratified epithelium, one of the functions is for protection, and keratinized stratified squamous is found in the epidermis and contains lots and lots of keratin, and that's for that protective purpose. Here is another slide. This tissue here, you can see that there's no apical surface. I don't see any really empty spaces, big empty spaces here to speak of. This one sometimes gets confused with blood because we do see these red dots. Those are the cells of this tissue, but I'm also seeing fibers. So no apical surface, cells that are spread apart, no lacunae, but I see these fibers that form this branching network. So all these blue or sort of dark blue, almost sometimes purplish fibers, those are forming this branching network, and our branching network is reticular. Reticular connective tissue forms stroma and binds smooth muscles together. It's found in the liver, the spleen, and the lymph nodes, and it's filled with reticular fibers. Okay, here's our next tissue type. This tissue does have an apical surface, so I'm looking here at these cells, and they come up next to an empty white space, so here's my apical surface. Here's my basement membrane down here, and if I look at that cell, it's very elongated and columnar in shape. There are cilia on the apical surface. These are my cilia, these hair-like extensions. You can also see them here, or down here, or over here. So because there are cilia, it has to be columnar. If I focus in on my nuclei, here are some nuclei here. Here are some over here. In most places on this slide, those nuclei seem to be all aligned in a row. So nucleus, 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 nucleus. They're fairly aligned in a row here. So it looks to me like a single row, simple, ciliated, columnar epithelium. Right? Our simples, they will not be for protection, but simples, columnar and simple cuboidal have the same functions, and that is secretion and absorption. For the simple ciliated columnar, we find this lining oviducts, lining the brain ventricles, and lining some of your bronchioles as well. Here's a pretty colorful tissue. This is one of the slides that we have for this particular tissue type, and it should look somewhat similar to the last slide in terms of the features we see. Big empty space here. We have that apical surface, so it's an epithelium. I also see cilia and a columnar shape here. So this is columnar epithelium. I also see goblet cells. So these sort of bubbly things in the middle of that epithelium, those are my goblet cells. If I look at the nuclei, here's one here, here's another, here's another, here's another, here's another, 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 another. The nuclei don't really align. They're somewhat irregularly spaced. And so this irregularity, this sort of random arrangement, tells us it must be pseudo-stratified, and it is ciliated, and it's columnar epithelium. Don't forget for columnar to put ciliated or non-ciliated for each of those types that you see. The goblet cells and cilia allow this tissue to trap and remove foreign material. It lines your upper respiratory tract and lines your trachea. All right, here's another tissue type. Let's look at this one. So once again, first thing, does that have an apical surface? I don't see an apical surface here. I see cell here, one there, one there. So no apical surface with my cells spread apart. I do not see lacunae, so there's no clear bubbles or empty pockets around these cells, so it's not cartilage. I see these thin fibers, and I also see thicker fibers scattered throughout as well. So this must be areolar connective tissue because areolar connective tissue has all three fiber types, and you will see collagen and elastic fibers on our areolar slide. The reticular fibers, remember, you won't see here because they need a special stain. Strength for the collagen fibers, elasticity for the elastic fibers, and support for the reticular fibers. We find this in your papillary layer of the dermis. That's the more superficial layer of dermis that's right underneath your keratinized stratified squamous. It surrounds organs and surrounds your blood vessels as well. So we see all three fiber types, reticular, elastic, and collagen fibers here. Next is another one that I think is pretty distinctive. We've got those tree rings. This is bone. Bone protects, stores, and provides for movement. The fibers that are in bone are collagen fibers. You don't see those super well here because they're embedded in this calcified matrix, but collagen fibers are contained in bone. We'll talk more about bone in our next unit. Next tissue type. So what I see here are all of these sort of circles. I see a circle there, a circle here, here, and here, with all of these lumens, these empty pockets in the middle. That's going to be my apical surface here. So my empty space with my apical surface. I see nuclei aligned in a single row. So apical surface, a single row of nuclei, and then a basement membrane here on the other side. Here's another example. Lumen, my apical surface, a single row of nuclei, and then that basement membrane. Those nuclei are nice and round, and these aren't squished, but they're also not drawn out and elongated, so they have a cuboidal shape to them. This is simple cuboidal epithelium, a single layer of cuboidal cells. Just like simple columnar, its functions are secretion and absorption. It lines your kidney tubules and covers your ovaries. All right, next one. If I look at this slide, I see really no white space to speak of, so no apical surface here. My cells seem to be stacked right next to each other. So no apical surface with tightly packed cells. This must be a muscle tissue. So my next muscle tissue question is whether or not there are striations. And you would be able to see these a little bit better looking at them in person. It didn't capture real well when I was taking the picture, but these are your striations, these stripes going perpendicular along the length of the cell. So those striations, along with the fact that my nuclei are pushed to the sides or edges, periphery of the cells, tells me this must be skeletal muscle. Skeletal muscle, like all other muscle, again, you just need to know muscle function in general, movement, heat generation, posture. All right, our next tissue type, we see an apical surface here. Here's my empty space. This must be my apical surface of cells all through here. And if I look at the shape of the cell, it's elongated, it's drawn out, so it is columnar. Looking carefully at the apical surface, although some areas look a little bit fuzzy, I don't see any distinctive hair-like extensions. So there are no cilia on that apical surface. So I know it's columnar and it's non-ciliated. The next question is, how many rows of cells do I have? If I look along these nuclei, they seem to be aligned mostly in a single row, with just a few exceptions of weird nuclei that are off-center. But for the most part, these are all kind of aligned in a row. So this is a single row of columnar cells with no cilia. Non-ciliated, simple columnar epithelium. Like our ciliated columnar epithelium, we also are going to use this for secretion and absorption, and it's found lining the digestive tract. All right, next tissue type. Let's take a look. I do have some empty spaces here, but if I look at where my cells are, they're here and here, over here, over here. The cells seem to be pretty spread out, so it must be some sort of a connective tissue. Next question is, do I have lacunae? And if I look at those cells, I can see that there's really not any pockets or bubbles around most of those cells. Okay, so it's not a cartilage. It's a connective tissue, but not a cartilage. And next thing I'm going to look at are, do I see fibers? And all of these red lines are fibers. And they're going in many different directions. So because the fibers are going this way, or this way, or this way, this must be an irregular arrangement of fibers that are bundled together. It is a dense, irregular connective tissue. Dense, irregular connective tissue provides tensile strength in multiple directions because the collagen fibers are running in multiple directions. It is your reticular layer of your dermis, that's the deeper layer of dermis. It also is found in joint capsules and in the fascia, and the relevant fiber here is collagen. All right, next tissue type. We're getting close to the end. Step 1, is there an apical surface? I see this big empty space. This would be my body cavity, outer covering, etc. So I must have an apical surface. This has got to be an epithelium. Step 2, how many layers of cells do I have? If I look from my apical surface until I get to my basement membrane, there are multiple layers of cells. So this must be a stratified epithelium. If I look closely at the apical surface, those cells that I see there are squished and flat, so this must be a squamous epithelium. So it is stratified squamous. Because I don't see that flaky outer layer, this is the non-keratinized stratified squamous epithelium. Like all of our other stratified epithelium, it serves to protect, and it's found lining the internal entryways to our body, lining the mouth, lining the esophagus, and lining the vagina. All right, next one. This tissue type, and I would be pointing you to this particular area on the slide here. So I'm going to see a lumen. This is my opening, so there is an apical surface here. Let's look at how my nuclei align. I see a layer of nuclei, and this is maybe a little bit better depiction actually. I see a layer of nuclei, and then I see another circular layer of nuclei. So I see 2 rows of circularly arranged nuclei. This must be stratified. And those cells are more cuboidal in shape, and it's really hard to see the boundaries of the cells, but because those nuclei are so nice and round, and because there are 2 short layers, this tells me it must be my cuboidal epithelium. So this is stratified cuboidal. Once again, it's stratified, so one of our functions is protection, and like our simple cuboidal, we also have secretion and absorption here too. This is found in your sweat glands, salivary glands, and a lining part of the urethra as well. All right, let's take a look at this one. This particular tissue type looks like a whole bunch of bubbles, okay? However, there is no apical surface here. There are no lacunae. There are no visible fibers. It basically looks like a whole bunch of bubbles. And if I look closely at those bubbles, I'll see maybe nuclei here and there kind of pushed off to the corner of those big bubbles. And that's what tells me this is adipose tissue. If this were simple squamous also kind of looks like bubbles, but the lining of those bubbles are all cells. It's all my squamous cells. We don't see that here. This is adipose tissue. Adipose tissue is basically fat, so it's going to provide insulation and energy reserves. It is found in the hypodermis layer of your integument. It surrounds the heart, it surrounds your kidneys, and it makes up your yellow bone marrow. All right. Next, we have this tissue type. So although again I see some sort of empty white spaces, there's no real apical surface to speak of. My cells are tightly packed, so I see cell next to cell next to cell. Here's a nuclei here. And my nuclei are in the middle of my cell. So if I look closely, I'll see striations. This tells me it's one of my muscles. Tightly packed cells with no apical surface also gives away that it's a muscle. And because we have striations and the nuclei are located in the center of that cell, that tells me that this must be cardiac muscle. And for cardiac muscle, we'll also see branching of those muscle cells. Like our other muscle cells, this functions for movement, generating heat, and for posture. And we have arrived at the last tissue. So, one more time, do we have an apical surface? Yes, we do. There's the empty space at the top with our apical surface of cells. So we know it's an epithelium. Do we have multiple layers of cells? Yes, we do. Multiple layers of nuclei aligned. So it's got to be a stratified. So you now might be thinking, what cell shape am I looking at? And this is that tricky one where it's not squamous, it's not cuboidal, and it's not columnar. If you look at that apical surface of cells, they are sort of dome-like in shape, and that is indicative of transitional epithelium. Transitional epithelium will change shape. That's the transition part. So when the bladder is empty, it has this characteristic shape. When the bladder is distended, it will have a flatter shape at that apical surface. So functions here are distension, and it lines your urinary bladder and your ureters. Okay, so let's recap. What you need to do. Step 1, identify the tissue type. Is it epithelium, connective, muscle, or nervous? And then use those tips and tricks we discussed to narrow down to a subcategory. Once you can identify all the tissue types, then you're going to want to work on functions. And a really useful thing to do here may be to group functions together. For example, we've got simple cuboidal, simple columnar, both ciliated and non-ciliated. Those all have the same functions of secretion and absorption. So making a chart might be helpful in you studying these different functions of these tissues. Finally, add in locations. The fewest number of questions will focus on locations, but there will still be a few. So add those in last, and then you'll have a complete picture of epithelial tissue. Oh, of all of our tissues, I should say. Don't forget there will still be a number of questions on anatomical terminology, so go back through your exercise 1 or through chapter 1 in your textbook. You need to know the directional terms, you'll need to know the locations on the body, your regions like buccal and vertebral and umbilical and popliteal. You'll also need to know your body planes and your body cavities. And don't forget to review the abdominopelvic regions and abdominopelvic quadrants. You'll need to be able to identify what those are called. Then the last thing is our integumentary system. There aren't a lot of terms here. There won't be a ton of questions, maybe 4 to 5 questions at most, but make sure that you can identify the structures that are on that integumentary system list. So review the slides that were presented this week in lab, and then also go through those model study guides so you can identify structures on the study guides. I'll have a few more reminders about the lab test going out in the announcements this week. They'll probably come out tomorrow morning, Saturday morning, so look for that. And then email me with your questions over the weekend. Happy studying, everybody. See you Tuesday.